welcome to another new episode video of BioBytes. Now, let's begin with a riddle. I am the energy maker inside you, powering everything you do. I take in food and oxygen and turn it into energy you can use. I work in millions within a cell, keeping life strong and running well. I have two membranes and my own DNA, a tiny factory just like a power plant. Who am I? By now you might have understood which cell organelle are we discussing. Yes, you are all right. We are talking none other than the powerhouse of the cell, the mitochondria. So mitochondria, they are the powerhouses of the cell because they are the ones which produce energy, and because they produce energy, every function of the cell continues, it's maintained, and it is mainly responsible for cellular respiration. As we know, respiration is nothing but where energy is produced and food is getting oxidized, and it has its own DNA and ribosomes. Mitochondria and plastids, these are the only two cell organelles which have their own genetic material, and because they produce energy and support all the functions of the cell, they help in growth, repair and survival. So mitochondria, they are essential for all life and all cell activities. They are the cell's power factory, and they convert the food into usable energy in the form of ATP. So let us now learn about the powerhouse of the cell, that is a mitochondria. Now let's talk about the history of mitochondria. Right. So if you just see this term mitochondria, mitos, which means thread, and chondrion means granule. Right. So they were first observed in the form of thread. They are thread-like granules. So they were given the name mitochondria, and they were first observed by Colliker, and he gave the name as sarcosomes. Mitochondria were first named as sarcosomes. Later, Altman, another scientist, thought they are living organisms, and Benda gave the name as mitochondria. So for the first time, the term mitochondria was given by a scientist called Benda, and Potter and Palade described mitochondria as cells inside cells. So we will be learning why they are called as cells inside cells. Let me tell you about a theory called as endosymbiotic theory. So this is a very important theory related to mitochondria, which scientists say it is called as endosymbiotic theory. Endosymbiotic theory. Now, you are all aware of what symbiosis is. Symbiosis is nothing but give and take relationship. So, children, scientists believed that once upon a time, these mitochondria, they were some living organisms which entered into the cell. So, they entered into the cell, and then later on, when they entered into the cell, these mitochondria provided energy to the cell, and the cell gave them protection and all the necessary things for their survival. And later on, after millions of years, the mitochondria became a part of that particular cell. So that is what the endosymbiotic theory supports, cells inside cells. Right. Now, occurrence, number and shape. Now when you see mitochondria are absent in prokaryotes and they are absent in mature mammalian RBC, they are present in large numbers in eukaryotic cells. Now, if you see the different shapes of mitochondria, some are spherical, some are granular, some are rod-like, and some are filamentous or thread-like shape. They vary in their shape. Now, if you see the number of mitochondria, there also is a variation. In a type of amoeba called chaos, you have nearly 50,000 mitochondria producing energy. In the egg of sea urchins, you have 150,000 mitochondria. In our liver cells, in every hepatocyte, we have 500 to 1600 mitochondria. right? Because the liver performs more than 500 functions, so it requires more energy, so it has more mitochondria. So these related to the number, related to the shape, this is all about the occurrence, number and shape of mitochondria. Now next, let us move on to the basic structure of mitochondria. Children, mitochondria are double membrane bound organelles. So they are double membrane. Till now we have learnt about endoplasmic reticulum, we have learnt about Golgi body, we have learnt about lysosomes. All these three, they were single membranous structures. But mitochondria, they are double membranous structures where you can see the outer membrane. The outer membrane is smooth and it is porous also. The outer membrane is smooth and porous to allow substances, easy passage of substances. Right? It is the outer boundary. The inner membrane, you can see this is the inner membrane. The inner membrane is highly folded. You can see how the inner membrane is highly folded. And these folds are called as cristae. So just remember, the outer membrane is not folded, it is porous. The inner membrane is greatly folded, highly folded, and those folds are called as cristae. Right? Now, the importance of these cristae is these cristae increase the surface area. You can see due to these folds, the surface area inside is increased. Therefore, more surface area, more energy production. Right? Then next, inside the cristae you have the substance, the matrix. Right? It is called as a matrix. It is a fluid filled space and it contains different types of enzymes like DNA, RNA, ribosomes and granules. Now if you see the inner membrane, inside the inside the matrix, in the matrix you find some structures called as F1 particles. Right? They are known as the F1 particles, or they are also known as the elementary particles or oxysomes. Oxysomes or F1 particles, which have a knob like structure, which have a stalk and a basal plate. Right. Now these elementary particles or racket particles, you also call them, they contain a special enzyme called as ATP synthase. Right? And this ATP synthase is mainly responsible for production of ATP, we will be learning. Now the distance between two F1 particles, so the whole of the matrix inside the matrix inside, it consists of these elementary particles with a knob, with a stalk and a basal plate, and between two F1 particles, the space between two F1 particles is 100 Armstrong units. Now the cristae, right, these cristae, the folded structures, they vary. So some mitochondria have parallel cristae. You can see they are parallel. Most of the muscle cells have this type of mitochondria. Most of our cells have irregular and branched cristae like this. You can see irregular cristae. Now in some of our cells we have spherical cristae. Right? You can see underdeveloped cristae, right, which are seen in less active or certain cell types, which are where the cristae are very few and small, and in some the cristae are absent. Mostly this type of mitochondria where cristae are absent, it is seen in some lower forms or some specific conditions where not much energy is required by those organisms. Right? So what all we have learned, you can see here, mitochondria are absent in prokaryotes, present in eukaryotes. The shape varies, right? The number varies. The outer membrane is porous and allows the passage. Inner membrane consists of cristae, and this cristae is the site of ATP synthesis. Right. Now let us learn about the structure, composition, enzymes and origin of mitochondria. Now as we have already discussed this, outer membrane, the inner membrane, the cristae and the matrix we have already discussed. Now if you see the chemical composition of mitochondria or the enzymes present in the mitochondria. So first let us talk about the enzymes present in the mitochondria. They are present in the outer membrane, they are present in the inner membrane, and they are present in the matrix. Matrix is the inside. Now if you see the outer membrane, let us just see that the outer membrane is the first floor, the inner membrane is the second floor, and the matrix is the third floor, right? The first floor, the second floor, and the third floor. Let us assume in this way. Now the outer membrane, you know, the first floor or the outside part, right, the security gate, right, the outer membrane acts as a security gate, so it allows the materials to enter or the check the incoming substances. So here you have the enzymes like monoamine oxidase, NADH, cytochrome, right, hexokinase, so the acetyl coenzyme A. So these are all the enzymes which are related to allowing the materials to come in. So these are all the enzymes. They act like the factory's reception or at the security gate. So they help prepare and admit the raw materials. Now let us talk about the the inner membrane. So the inner membrane acts as the power generation hall. So it is very important floor, right? The inner membrane, it consists of very important enzymes like NAD, FAD, right, DPN, all the four types of cytochromes, coenzymes, ATP synthase, and succinate dehydrogenase. So all these here you see. Where is electricity produced in a power station, children? Generally it is produced in the generator room. So exactly, the inner membrane acts as a generator room, right? So it is the room where electricity or where the power is produced, ATP is produced. Here everything is involved like energy transfer, electron transport, ATP production, all these occurs here, right? Now if you see the third, right, so the third one, the inner membrane, the matrix or the M phase. So here inside you have we call it as a food processing unit, right? So here you have the enzymes like fumarase, aconitase, malate, isocitrate. So these are the enzymes, right, which like you see, they mainly convert the energy present in the food into usable energy. So imagine it is like a relay race. So runner 1, runner 2, and runner 3, right? So these runners are NAD, FAD, right, coenzymes, right, cytochromes. These are all like relay runners. So we will not learn more about in detail about these enzymes. So the outer membrane is the entry gate, the power generation hall, and the processing and the separation unit. So these are all the enzymes related to this. Right. Now, children, let me tell you all the main function of mitochondria, that is a glycolysis, Krebs cycle, and electron transport chain. We will be learning in a separate class. Right. So now the other than these three, we will be learning now. Now, if you see the functions of mitochondria, it is a site of oxidation, right? Dehydrogenation, phosphorylation, production of energy here, right? It is a site of ATP synthesis. It is a main respiratory organelle, that is where food is oxidized and energy is released. It is associated with fat metabolism, breakdown of fat molecules, right? It is associated in the formation of sphingolipids with the Golgi body, right? The way Golgi body form lipids, right? The main organelle involved here is mitochondria. Now let us talk about the arrangement of in the inner membrane. Here what you see, children, here this is nothing but this is the arrangement of the mitochondrial membrane. Now if you see this mitochondrial membrane, you can see this is the intermembrane space and it is a matrix. So this part is the matrix part, this part is the outer part. Now you can see these are the protons, right, which have to be passed. So it is like a power generation wall, right? So imagine this is like a power generation wall. Many enzymes and proteins are embedded in this wall, and together they help in conversion of food into usable energy. So if you see the intermembrane space, that means the space which is present between the outer and the inner membrane, this is the intermembrane space. Think of a corridor. So it is like a corridor between the two walls. Right. Now if you see in the M phase, matrix phase, it contains DNA, it contains RNA, right? It contains ribosomes, enzymes. So it is like a control room of mitochondria where ATP is produced. Now, in addition to this, you will see there are some, there are some like NADH. You can see energy relay runners. There are some type of energy relay runners, I should say, like NADH. So NADH is like a charged battery carrying energy. It is the one which is very much significant for the production of energy. It delivers the energy to the membrane proteins, and you can see the non-heme iron, right? So these are iron containing proteins which help in transfer of electrons, right? That means they are involved in energy transfer. And you can see there are some protons here, right? So these protons are nothing but these act as pumps, right? So the proteins pump these protons across the membrane here, right? So you can see one more very important structure like a mushroom, a very important structure in the diagram here. We call it as ATP synthase. It is also called as, very important to remember, ATPase or ATP synthase. It is also called as F1 particle or ATP synthase. Sorry, my pen is troubling me. It is also called as ATP synthase, F1 particle, or it is also known as oxysome. This mushroom like structure, the ATPase. Its job is, the main job of this is, it takes up the, you can see here, for the ADP, adenosine diphosphate, it adds the inorganic phosphate and converts it into ATP. That is done by mostly the ATPase, right? It acts as a microscopic turbine. So this ATPase acts as a microscopic turbine that manufactures ATP, right? You can see the energy currency of the cell, right? In addition to this, it is like nuts and bolts holding the ATP machine together, right? So our NADH, in short, our NADH is like the fuel supply, the protein complexes are like the pumps, right? And the H plus ions are like the water stored behind a dam. The ATP synthase is like a turbine, and ATP is the electricity. So it is like a miniature like power station built inside the inner membrane of mitochondria, right? Now if you see the origin of mitochondria. So the mitochondria the scientist by the name Novikov said that mitochondria arise from nothing, without, there is no pre-existing substance, it just arose just like that. But a pro-mitochondrial theory says that there were some small particles in the cytoplasm called as pro-mitochondria, right, which continued to grow, and they gave rise to mitochondria. That is a pro-mitochondrial theory. There is one more theory, Tandler and Luck theory, which says that new mitochondria are produced by existing mitochondria, right, from the septa. There is also another theory, Morrison theory, which says that mitochondria arise from the plasma membrane, from the endoplasmic reticulum, either from the plasma membrane or from the ER, right? So these are the theories which were put forward related to the origin of mitochondria, right? So this is all the chemical composition of mitochondria, which contains these proteins, phospholipids, right? And you see children how I told you that about the the theory, the endosymbiotic theory, cells inside cells. So what is the evidence behind that? How can we say that once upon a time mitochondria were separate cells which entered into another cell and this became a part of it? Because they have the 70s ribosomes, right? Because they have the 70s ribosomes, and they have some DNA and RNA in them. So these are very strong evidences which prove the endosymbiotic theory, right? Now it's time to test your understanding. So in our class we have learned only the the gist of mitochondria. In our next class we will be learning in detail about glycolysis, Krebs cycle and electron transport chain. Now let us talk about this. A scientist observed an organelle with the following characteristics: the double membrane bound, right, circular DNA, 70s ribosomes. Which theory best supports? So it is very easy. Endosymbiotic theory, which says that once upon a time mitochondria will like were like separate cells which entered into another cell, right? And then they became they provided energy to this cell, and in turn the cell gave them support. So like that they became a part of this cell. So the endosymbiotic theory supports this, and the evidence is circular DNA, 70s ribosomes. Now a mutation causes complete disappearance of cristae. So there is complete disappearance of cristae from mitochondria. So cristae are nothing but the inner foldings of mitochondria. We call them as cristae. So which process would be mostly affected? So if cristae are absent, the ATP production will not take place. So the ATP synthesis associated with the inner membrane complexes will get affected. Then next, which of the following combinations is incorrect? So outer membrane is smooth and porous, right? This is correct. Matrix contains DNA and ribosomes, correct. Cristae are formed by the outer membrane. Remember, outer membrane, cristae are not formed. Outer membrane is just it doesn't have any folding. So this seems to be wrong. F1 particles are associated with ATP synthesis. So the incorrect one is cristae formed by the outer membrane. No, they are formed by the inner membrane. So the answer is C. Now let us match this. Oxysomes. So oxysomes are ATP synthetase particles. So oxysomes, they are ATP synthetase particles, correct. Matrix. So matrix is the one. Okay, so let us talk about cristae first. Cristae is nothing but they help in surface increasing the surface area. The outer membrane is porous. Very good. And the matrix is nothing but it is a like fluid filled space which is rich in enzyme rich cavity. So the answer is A, right? 1A, 2C, 3D. So this is the column matching with column B, right? Now this is something very interesting. A researcher compares two cell types. First cell contains very few mitochondria, so low number of cristae, and cell Y contains innumerable mitochondria or numerous mitochondria and highly folded cristae. Which statement is likely correct or most likely correct? Cell A has high ATP demand. So this is not possible because mitochondria are very few. So demand of ATP? No. So this is not correct. Cell Y has higher metabolic activity because many mitochondria, many folded cristae, lot of energy is released, lot of ATP is released. When lot of ATP is released, naturally metabolic activity will be high. So let us hold it. Cell X is a muscle cell. In our muscle cells, energy required should be more. So more mitochondria. So this is not applicable. Both cells have equal ATP production. No, few mitochondria, numerous mitochondria, ATP production will vary. So the correct answer is cell Y has higher metabolic activity because it has numerous mitochondria. Now, a mitochondrion is found to contain circular DNA, RNA, 70S, ribosomes, ATP synthase, respiratory enzymes. Which characteristic most strongly supports the idea that mitochondria were once free living organisms? How can you say that mitochondria were free living organisms? Yes, presence of circular DNA and 70S ribosomes. Already it is given here, right? So it is, the answer is C. So this is all with mitochondria. I'll come back with another function of mitochondria with glycolysis, Krebs cycle and electron transport chain all together. Keep learning, enjoy and happy learning. Thank you.